everyone. Welcome to the fifth version of the NOAHE Rounds series. This is our first session in 2021 for this, or 2021-2022 academic year. I'd like you all to please join me in welcoming our presenter, Dr. Anna Heath, who is an assistant professor at the University of Toronto and a scientist at SickKids and an honorary research fellow at University College London. I first met Anna while we were both based at UCL, where she completed her PhD in 2018. She worked under the supervision of Gianluca Baio, and her thesis was Bayesian computations for value of information measures using Gaussian processes, INLA, and moments matching. A very technical take on things that at least we're very interested here at IHE. She also published a book with Gianluca on Bayesian cost effectiveness analysis with the R package, BCEA, which is a really helpful guide to doing the kinds of work that many of us here on this call do using R and the BCEA package, and I recommend you check that out. More recently, Anna's been working with us at IHE on a suite of studies through the Innovative Pediatric Clinical Trials group, which has been really interesting. She somewhat left us to go take up her assistant professor post. So we do miss her, but glad to have her back with us today to talk about this. So her talk today is going to be on designing clinical research to support decision-making, a comparison of methods for value of information. And just before I hand over to Anna, I just want to go through a couple of housekeeping items. Participants will be muted throughout the webinar, so just so you're aware of that. If you'd like to submit any questions during the webinar, please use the Q&A function. And then we will facilitate questions that are in the Q&A space at the end of the webinar, okay? So thank you again, Anna. I will now hand over to Dr. Heath.
Fantastic. Thanks, Jeff. So I'm really happy to be here today, and thank you very much for the invitation. I'm hoping to kind of just give a brief introduction to value of information for those of you who maybe have heard of it, but maybe aren't so confident or haven't heard of it before. And then I'm going to move into kind of talking about some of the challenges of value of information and maybe why it's not being used so often. And then some of the work that I've been doing along with a large number of collaborators in the collaborative network for value of information, all the work that we've done to try and kind of understand how these different kind of solutions to the challenges that we see in value of information, how they kind of work together and how we can help people really navigate the space and do value of information in practice, which is kind of the overall goal of what we aim to do. So I'm just going to start. I now work in a hospital, so a lot of my kind of work is on trying to kind of work out how to take these methods, which are very relatively theoretical, have mostly only been used in kind of theoretical terms, and how to translate them into practice. So I find that the best kind of way to start is essentially value of information methods are kind of a method for research design. So before we kind of dive into that, just wanted to kind of briefly talk about how we kind of currently do research design and what I mean by research design. So in general, I think research design kind of takes two kind of key parts. The first is just the prioritization phase. So what's actually the key goal of doing the research? What do we actually want to investigate? And traditionally those priorities are kind of informed by the literature, but they're also informed by stakeholder groups or experts that kind of get together and maybe identify the key kind of missing pieces in the literature. And usually, particularly in my work, when we're talking about evidence and missing gaps, we're talking about randomized control trials. Like, where do we need another randomized control trial in order to understand, what the best treatment is? And traditionally, what we would then do is kind of, again, with looking at stakeholders or talking to stakeholders, we would maybe decide on the specific hypotheses or the specific outcomes that are maybe relevant to the this research, the kind of overall research priority that we've identified. And then we would design kind of an experimental late stage trial, particularly in the work that I do, that aims to show statistical significance for the primary outcome of interest. So that would be, trying to find potentially the prioritization would be trying to work out what the appropriate kind of pain medication is for children in the emergency department. And then we would narrow that down. And the primary outcome would be kind of pain at 60 minutes after enrollment in the trial. And we're going to, show statistically significant improvement for a combination therapy versus a monotherapy. We would then undertake that study, and kind of in a cycle of evidence way, we'd expect that RCT to be included in kind of future systematic reviews and kind of feed into the research prioritization and study design in an informal way going forward. But kind of the issue with that is that there's a kind of translation gap. So often these RCTs that we design that are very expensive, they don't really translate into clinical practice and changing clinical practice. And one of the reasons for that is that clinical practice is influenced by policymakers. Now, I'm sure, again, this is preaching to the choir a little bit here, but policymakers don't just care about RCTs, they care about cost, they care about quality of life measures, and all sorts of other things. And they're also, their decision-making that tries to combine all of these pieces of evidence on efficacy, costs, quality of life, etc., is often supported by a decision model. So we're building a decision model to understand or evaluate the net benefit of the technologies that we're assessing. And then that would then feed into clinical practice. So those guidelines are also really an important part of making things translate into practice. And we would always, when we're doing this kind of analysis, is kind of consider decision uncertainty. So how does the uncertainty in the evidence, i.e. the uncertainty in the cost or the quality of life or the randomized control trial data that we have, how does that impact our ability to make decisions? So feeding that into the decision model through a probabilistic sensitivity analysis, if that's something you've done, how does that impact our decision making? So because we have this kind of disconnect, and I don't under any circumstances claim that I'm going to solve all those problems, because of this disconnect and some other kind of key elements, there's a huge amount of research waste in clinical research. So I think in 2009, it was estimated that 85% of funded clinical research does not go forward to affect practice or patient care. And that's actually recently been updated, I think in 2018, and the estimate was very similar. And some of these are to do with bias or underreported studies, inaccessible research information, which I don't claim value of information is going to help in any way. But the two kind of key things that it can really help with is making sure that the research is relevant to the users of research. So understanding that policymakers and decision makers are a user of research and trying to take into account their perspective when designing the study. And also making sure that our designs, methods, and analysis are relevant to those decision makers, but also, accurately put together. So all patients are disadvantaged from this lack of translation, this kind of disconnect between research prioritization, study design, and the decision-making process. Those that are in the trial are kind of, put in lots of effort and don't really see the benefit that they're expecting. And those patients that don't receive effective treatments because of a translation gap are also disadvantaged. And there's huge sums of public funding that are used inefficiently in biomedical research. So really thinking about how we can kind of quantify the value of research and make sure that we're maximizing that value is a really kind of natural thing to think about in this framework. And value of information is a potential way to do that. So I like to think of value of information as something that kind of interrupts this cycle. So it takes the evidence and it feeds it into a decision model. And from that, we then undertake a value of information analysis that allows us to undertake research prioritization and study design to kind of work out what's the best study, what's the studies that we should be prioritizing in order to help make clinical or policy decisions that are based on decision models. We then would kind of undertake that study and feed it back into the body of evidence before kind of redoing our decision analysis and working out potentially again, using value of information, whether there's kind of additional research that needs to be done, and potentially going around this cycle multiple times before we make a decision and kind of go forward to clinical practice. But at each point, we're really taking into account the fact that the policymakers are going to be using these models to make their decisions. And we really need to make sure we've got the right evidence in the right place to make those decisions. So just in general, value of information analyses can answer kind of the 3 following questions, or at least that's how I conceptualize it. So firstly, they can answer, do we require any further research, or is the currently available evidence sufficient to determine health policy? So before we even enter into the realm of what research we should do or what outcomes we should measure, how we should design it, we're just asking ourselves a really basic question, like, do we even need any research to start with? Then they can answer the question, what measures or outcomes should we prioritize to accurately guide practice? So do we need to know something about the prevalence of the disease, the success rate of the novel intervention, the costs of treating the side effects, the quality of life measures going forward after treatment? Like, all of these things are potential outcomes of a study. And it's really important that we make sure we're targeting the outcomes that are going to give us the biggest benefit in terms of supporting our decision making. And then finally, we can actually ask, how should we design this study to collect the additional evidence? So do we actually need a clinical trial? And if we do, what's the sample size of that trial? What treatments, what interventions should we have in that trial? Or maybe we could do a registry study or use, observational data. And if that's the case, what outcomes do we really need from that study? How many patients do we need? And all of these questions. So we're really talking about a kind of overriding prioritization and design framework that addresses both of those key concerns. So before we kind of dive into the technicalities and the complexities, I just wanted to give this kind of simple toy example to really understand, or to drill home, like, why does information actually have value in the first place? Like, we're talking about value of information, but what does it actually mean? So I'm just focusing on a really simple decision model. So this is, I guess, a 2-stage, a 2-decision case. You could either basically, you have a patient who's arrived at kind of a doctor's office, and the goal is to determine whether you're going to prescribe antibiotics to that patient or not. And what we have is we have our kind of outcomes. So if you're looking at the top of the screen, you'll see that the first decision, I guess, is to prescribe antibiotics. And if you prescribe antibiotics, the patient will either have a bacterial infection, in which case their bacterial infection will be cured by the antibiotics that you've given. And the cost of that, the cost of giving antibiotics is $5. That's the cost of the drug. And because you've got no adverse health outcomes from prescribing, you would just give a cost of $5. However, if the patient didn't have a bacterial infection, you've kind of incorrectly prescribed antibiotics, and we're going to associate that with a cost that we kind of assume to be like the individual level cost of contributing to antimicrobial resistance going forward. In that case, we've decided that cost comes to $200. Or the other option would be to not prescribe antibiotics. And if the patient didn't have a bacterial infection, then there'd be no cost. You didn't prescribe the drug, you haven't contributed to antimicrobial resistance. However, if they do, you have a cost of $5,000, which is the cost of emergency hospitalization to treat the side effects, or the disease going forward. So we need to ask ourselves, which decision should we make? And the key piece of information you kind of need there is, what's the chance of having a bacterial infection and what's the chance of not having a bacterial infection? So in this case, we're going to calculate the expected cost of each treatment arm. And because there's a 20% chance of having a bacterial infection in this very nice scenario, the expected cost of not prescribing is $1,000. So 20% of the time it will cost you $5,000, 80% of the time zero. And the expected cost of prescribing is $161. So at this moment, we should go and we should prescribe antibiotics to everybody because it's the best scenario in terms of minimizing expected cost. Now, the question we can ask ourselves is, rather than kind of making decisions in expectation, what if we knew whether the patient had a bacterial infection or not? So in that case, we could actually prescribe the correct treatment in both cases. So we've got this new test, and we're going to say that if the test is positive, we're going to prescribe, and if the test is negative, we're going to not prescribe. We assume it's a perfect test. So in that case, we've taken the test and we spend $5 when there is a bacterial infection and $0 where there isn't. So then to calculate kind of the expected cost of that strategy of testing and then prescribing, you just need to take into account that 20% of the time you'll have a positive test and 80% of the time you have a negative test. So the expected cost of this strategy is only $5 because it's just, no, $1, sorry, because it's $5 20% of the time. Of course, the other cost that we're forgetting here is the cost of the drug itself. So no, sorry, the cost of the test. So the cost of testing whether you have a bacterial infection or not, which essentially is the cost of obtaining information about the bacterial status of the patient. So how much should we pay for that test? And the answer to that is basically the difference between the cost that we had without the information and the cost that we had with the information. So we know that we're going to spend $1, on average every time we have the test, and we're going to spend $161 on average every time we don't have the test. So the value of learning that information is $160. So in that case, if the cost of the test was more than $160, we should just prescribe antibiotics all the time. But if the cost of the test is less than $160, we would take the test and then calculate the value. And essentially the reason here is that getting the information allows you to avoid the negative consequence of prescribing when you don't have a bacterial infection. So this is where kind of the value of information comes from. It comes from the fact that you kind of prevent yourself from making the wrong call by gaining the information. And it's the expected gain in value across all the two outcomes of the test. So how does that kind of translate into the much more complex setting of health policy decision making? So in this case, instead of having a kind of nice decision tree where we know that the 20% of the time we have bacterial infection, 80% of the time we don't, the uncertainties in our decision making come from our uncertainties in our model inputs. So that would be our quality of life, our costs, our treatment effects, which are estimated from data. We represent our uncertainty in those inputs using distributions. And it's the uncertainty in those inputs that we kind of feed into the decision model that gives us our uncertainty in our kind of net benefit or our cost, as we had in the example, of the two different treatment options. And typically we kind of understand this uncertainty rather than kind of doing everything kind of analytically where we could write out all the formulas. We do it by simulation. So we simulate where the pink crosses are in those model input distributions. We feed that through the decision model and we get a value for the net benefit of the first treatment and the net benefit of the second treatment. And we kind of continue to do this. So we repeatedly kind of resample from these distributions and we get values for the net benefit for treatment 1 and the net benefit for treatment 2. And we just keep going, we do this 1,000 times, 10,000 times, and that gives us a kind of representation of the uncertainty or our kind of distribution of the net benefit for the two treatments. But the key point for value of information is actually this pink line that I've highlighted here. So if we take the expected, the average net benefit, then we should always implement that second treatment with a value of $5,046. But there are some cases where you might make the wrong decision if you were to implement that second treatment there. And in the pink line, what you can see is that it's in this line that the net benefit for treatment 1 is actually higher than the net benefit for treatment 2. So it's for that pink line is where we've kind of made the wrong decision. And so by gaining additional information, by having kind of doing a study and understanding, we'll potentially be able to identify that whether we're in this kind of standard like state of the world where the second treatment's better, or by getting more information, we'd be able to say, oh, actually, you know what, we were wrong. And we're actually in this state of the world that we see from the pink line where the treatment 1 is better. So the information basically allows you to kind of make a better decision because it may indicate whether you're in this kind of state of the world where you've made the wrong decision. And the value of like finding out that you're in the wrong state of the world is basically the difference between the value of the two treatments. So it's kind of 0 when you make the right decision, and it's positive when you make the wrong decision, and the information would make you switch your decision. So I guess that's like a whistle-stop tour of value of information and where the information value kind of comes from. But really, when we're working in trials, when we're working in study design, what we're asking ourselves is kind of, should we collect additional data? And I like to think about it like this, I guess, again, like in a kind of decision tree kind of framework. Usually when we're asking in a kind of health policy decision-making context, we'd be just saying, which is the best treatment 1 or treatment 2? And we'd make that decision by maximizing expected benefit, like we did in the previous setting. And the expected benefit comes from this current level of uncertainty, so this uncertainty that we have. But value of information kind of asks you to make an additional to ask an additional question, which is like, do we accept that this current evidence is sufficient for decision making? Or do we need to buy additional information, which would lead us to an updated level of uncertainty? And actually, once we had that updated level of uncertainty, we'd actually make the same decision again. So we need to decide between treatment 1 and treatment 2, and we would do that by maximizing expected benefit. And actually, we can kind of think about this as a decision tree going off to infinity, where at each point where we kind of get our updated level of uncertainty, we're asking ourselves again, do we accept the updated evidence, or do we need to buy additional information? And again, once you get to the next node down, you say, should I accept or buy additional information? And that you would make the decision again. And the key quantity that kind of goes into that accept current evidence or buy additional information decision is something called the expected value of sample information. So the expected value of sample information estimates the value of reducing your uncertainty from the current level of uncertainty to the updated level of uncertainty. And if we can estimate the value of doing that, then we can compare that with the cost of buying the information. So when we talk about buying the information, we say the cost of the trial that we're going to do, or the cost of the study, the analyst time for analyzing the registry, or, whatever. We're basically comparing the value of the information to the cost of obtaining it. And that will basically give you a yes/no decision as to whether you should accept the current evidence or buy additional information. So if the current evidence, if the cost or the net benefit of the additional information is lower than 0, then we should accept the current evidence. And if it's higher than 0, we should buy additional information and make the decision in the future. So that's basically the goal of kind of EVSI. And that's how it kind of really can help you with trial design, because it allows you to really compute the value of what you're actually going to do and compare it with the cost of doing that research. However, there are huge kind of computational issues with calculating EVSI. And the reason for that is kind of the way that it's defined. So what we're saying when we calculate an EBSI is we basically generate kind of fake or hypothetical data. So in this case, represented by that kind of matrix. So this is saying we're collecting data from, I don't know how many it is, like 20 patients, and we're collecting 3 data points for each patient. And we're using those 3 data points for each patient to update the distributions of our uncertainty and our model parameters. And because we get an updated distribution, what we've got is we've got like a thinner distribution, and it's usually, or typically you have a thinner distribution, but it can also change its location. So that might mean that this particular data shows that the treatment effect is actually negative, whereas on average it's positive. And once you've got the data and the updated distributions for your uncertainty, you actually feed that through the decision model and you maximize expected benefit. So for those of you who run PSAs like normally, this is basically doing a whole PSA and maximizing expected benefit. But in order to calculate the expected value of sample information, this is essentially the value of sample information, but we want to do an expected value over all possible data sets. So we actually have to resample our data, redetermine the updated evidence, and feed that through our decision model, and then maximize expected benefit. So what we're doing here, rather than just one PSA, is we're doing, say, 10,000 PSAs. So we're repeatedly resampling that data, repeatedly combining it, and then feeding it through the decision model to maximize expected benefit. And so I think there's kind of, it's very obvious that this becomes like almost infeasibly like expensive or computationally expensive, even if you just have like a relatively simple model, like a Markov model. Even then, if you're trying to rerun the PSA of your Markov model, say 10,000 times, you're already looking at something that's going to take like a day of computation, potentially more. And in order to do trial design, so not just like we've decided what trial we're going to do, is it worthwhile or not, but actually what's the best trial that we can do to address these questions, you actually have to kind of think about sampling different types of data. So in this case, you could think about, okay, well, maybe I had a clinical, maybe gray represents a clinical trial with 100 patients, and yellow would be a clinical trial with 200 patients. So you're actually having to kind of, not just for each like study to calculate the EBSI would maybe take a day, but you're actually having to do it for multiple different study designs. And for each time, you're having to design the trial, generate the data, feed it in, do a PSA, and then do this repeatedly. And again, for another study design and another kind of key thing. So this, I think, becomes pretty obvious that this is almost an impossible task and something that people just don't have time for, especially in a relatively, trial design is often a relatively underfunded, but also time poor situation. You need the design like 2 weeks ago and you need it now. So this is something that just completely was a barrier to being able to do this and to being able to ask these questions in an intelligent way. So recently, there have been kind of 4 methods that were developed to overcome this computational barrier. And they were pretty much developed concurrently, although you can see from the dates, there's like 2015 to 2018, but they were all kind of developed as part of this kind of push to make value of information more easier to understand and to do. And so as part of the work that I've been doing, we've been trying to kind of make these methods easier to understand. Now, I think you'll probably be glad to hear that I'm not actually going to go through the technicalities of any of these methods, because I think they could each have their own hour-long presentation. And I didn't really think that was a good use of time. But just to kind of let you know, I'm going to use the same colors throughout. The four methods are what we've named the regression-based method, which was developed by Mark Strong in 2015. And then the importance sampling method, which was developed by Nick Menzies in 2016. The Gaussian approximation method, which was developed by Jalal and Alarid Escudero, building on some previous work that also used the Gaussian approximation, but wasn't as widely applicable as the kind of most recent iteration of the method. And then the moment matching method, which I developed as part of my PhD in 2018 and 2019. So the really like going into this space, there's these four methods. They were all kind of developed at the same time. They're all based on kind of different ideas and different ways of thinking. The key question that I think really jumps out is like, which method is the best? If we have these four competing methods, how are we going to kind of help people understand which one they should use? And as a statistician, the kind of classic answer to that is to do a simulation study. So to say, okay, great, I'm going to go and simulate all of these different methods. I'm going to compare them. I'm going to understand their characteristics, and I'm going to be able to come out with this key recommendation as to which one is the best method to use. But there's some huge kind of difficulties in this. And the first is that the performance of these different methods is based on the decision model that you use and what data you simulate. What's your like future proposed data study? So that already means that rather than being able to just say, okay, I'm going to fix like some key quantities, which is what you would do in a traditional simulation study, here we're not fixing like the mean of a distribution, we're fixing an entire decision model. And we can make that decision model almost as complex as we wanted or as simple as we wanted. But we're also fixing, you know, the data generating process, the study that we're interested in. So there's so many kind of pieces of this puzzle that actually makes it very difficult to set up this simulation study. But I think one of the other kind of key difficulties is what I like to call the conflicts of interest. So as I said, one of the methods was developed as part of my PhD. So it becomes very challenging to kind of do a fair comparison of these four different methods when you have four different teams that have all kind of developed them separately from each other. And it's all well and good to kind of come together and write a paper and say, yeah, great, mine's the best, everybody should use it. And like, but it obviously would be a pretty hard for people to maybe believe that that was the true outcome, and you'd end up potentially with a, you know, like people kind of competing against each other and all of that. So that was something we really wanted to kind of work with. Like, we didn't want to have something that really came as like a partisan paper comparing these. We really wanted to work together and get something that was actually going to be helpful for the community. Another kind of key problem, which ties in really closely with this conflicts of interest, is that the model performance is actually based on the choices that you make as an analyst. So I can design an exercise that means that, you know, the method that I developed maybe comes out on top, or very much comes out, you know, like looking more competitive than the others. But somebody else could do exactly the same thing and kind of think really carefully about their choices and kind of, you know, land on their method. And again, that wouldn't necessarily be helpful for people. And the final kind of real complexity is just the time required to get an accurate comparator. So when you do a simulation study, you usually want to know, like, what's the truth? But the truth here has to be estimated by that simulation-based method that we talked about. And I think as we go forward, you'll see one of the truths in this study actually took like 300 days of computation to obtain. And actually, one of the examples, we couldn't even get a truth just because of the complexity. I think it would have taken us something like 40 years to get the comparator. So we just didn't have the ability. So the way that we kind of tackled these issues, firstly, we kind of aimed to, rather than just have one example, we really wanted to have 3 case studies or multiple case studies that kind of tackle different types of decision models and different types of trials. So in this particular case, we evaluated, firstly, a novel chemotherapy treatment that reduces treatment-related adverse events using a decision tree coupled with a 4-state Markov model. And this is a toy example that I developed, but that's been used in several different papers to kind of explore value of information methods. And this had a proposed trial that collects information on 6 model inputs. So it wasn't just like a simple 2-arm trial. We had information about the length of time that you had adverse events, the quality of life of the adverse events, the transition probabilities within hospital to death. So it kind of had a lot of information. And these model inputs were also kind of related to each other. So, you know, it was talking about transition probabilities, and we had like rates of change and kind of survival times that went into a Markov model. Secondly, we wanted to evaluate 2 drugs to treat. So this was a model that was developed in practice by Sullivan et al. They actually provided the R code to myself while I was working on this. So this was actually a model that wasn't designed to kind of showcase value of information. And I felt that was a really important thing to kind of use a practical example. And this example, the proposed trial, aimed to collect information on quality of life. So rather than kind of thinking about it in a trial perspective, this was more of like a survey data and how did we kind of do quality of life data. And we actually assumed missingness in the data collection so that we would have a kind of lower survey response than the number of people that we actually investigated. And we evaluated the EVSI across 5 different sample sizes. So surveys between 25 and 150 patients. 150 kind of respondents. And then finally, we did a case study that evaluated a screening strategy for colorectal cancer using a 9-state Markov model, which with age-dependent transition intensities. And again, this model was developed by one of my collaborators, and they developed it kind of in practice with real data. So again, it was more of a practical example. And the proposed study was a cross-sectional observational study to understand the age-dependent presence of colorectal cancer in the population. So again, not a trial, and thinking about how we generated data from that observational study. And we evaluated the EVSI across 9 different sample sizes in that example. So in order to kind of tackle this conflicts of interest issue, we actually developed, and I spoke a little bit about this at the beginning, this collaborative network on value of information. So this is actually a slightly outdated version of our webpage. We actually have 20 kind of collaborators now from, I think it's 7 different countries, who are all kind of working together to try and improve the use of value of information in practice. And we have a couple of tutorial papers that have come out recently, and we do courses and things like that. So really trying to work in all those different spaces. But the first piece of work that we did was this methods comparison. And I just want to draw your attention, I guess, to the fact that we actually had representatives from every kind of methodological innovation paper. So Hawe Jalal and Fernando Alarid Escudero. We also worked with Mark Strong and Nick Menzies from Sheffield, alongside other kind of co-authors on those papers. And we really, we wanted to bring everybody together and everybody to kind of give their support on how we were going to do this analysis and comparison to really make sure that it fairly compared the methods as much as we could. And when I'm talking about making kind of fair choices, the real challenge with this project was that as in all statistics, the higher the sample size, and by this I mean kind of like simulation size, the higher the simulation size, the higher the computational cost, but also the higher the accuracy. Now, the problem with that is that we have these 4 different methods. And actually, what I'm hoping to demonstrate in this plot is that the relationship between sample size and accuracy is different for each of the methods that we have. And so what that means is that you can kind of manipulate the choices that you make to choose kind of any part of this plot where one of the methods is superior, if that was what you really wanted to go out and achieve. So for example, if you wanted to really think about the pink method, you would choose smaller sample sizes, whereas if you wanted to kind of improve the purple method, you would choose larger sample sizes and really show the difference. So we tried to make fair choices, and by that I mean we tried to make kind of representative choices of how people might do value of information in the real world. And then finally, to make sure that we had an accurate comparator, we actually used lots of high performance computing. So we just tried to overcome those barriers by kind of EVSI can be easily parallelizable, and we kind of fed it to the supercomputer. We waited for a month to get our 300 days of computation, and we took those results back. But even with high performance computing, we really did have a limited number of comparisons that we could do to allow for feasible results. So we tried again to represent, to kind of work with the team to represent as fairly as we could the limited comparisons that we had. So just to give you an idea of the results, in the chemotherapy example, so the first case study, you can see that the regression-based method, the purple line, is by far the best method. It has the smallest kind of uncertainty. So this was done by repeatedly recalculating the EVSI. And you can see that it has very small like bands in terms of uncertainty, and also that it's really close to the true EVSI, which is represented by the black line. The moment matching method has kind of huge uncertainty in terms of huge variation in terms of results. And the other two are kind of somewhere in the middle, but are obviously kind of biased, like they were always higher than the true EVSI. In this example, in the chronic pain example, we have the black lines again represent the kind of true value, and the dots are the same methods as before. So in this case, the moment matching method seems to be kind of more accurate than the others. But again, the regression-based method has much smaller uncertainty, so it's much less likely to be kind of varied. But the other two methods, the importance sampling, Gaussian approximation, are much closer in this setting, although the Gaussian approximation does start to become downwardly biased as we go up in sample size. And what's actually interesting, I guess, is that in the first example, these are upwardly biased, and in the second example, they're downwardly biased. So there doesn't seem to be any kind of systematic bias that we could adjust for. And then finally, we did the screening example for colorectal cancer. The challenge was this, that we weren't able to get the truth. So what we were really looking for was, do the estimates kind of, are they close to each other, at least to give some indication that they're estimating the same thing? And then the other thing to say is that the black line at the top is the maximum, is the EV PPI, which represents the maximum value that you can get as an EVSI. And we see here that the importance sampling method is above that line. So there are some kind of estimation errors. And finally, for the largest sample size here, the importance sampling method actually has huge computational issues, and it means that we actually get a nonsense estimate, essentially. So what we did kind of find out from this was that we wouldn't recommend the importance sampling method for large sample sizes, just because of the computational issues that you encounter. But other than that, we have kind of relatively good confidence that these methods are all estimating the same thing. When it goes down to kind of computational time, so the amount of time and effort that it took to get these approximations, you can see that it's highly variable depending on the example and the method. So the regression-based method is pretty small, irrespective, so about half a minute for the colorectal cancer and shorter for the others. Whereas the importance sampling method kind of starts really low for the chemotherapy example, but gets much more expensive as you go higher. The moment matching method actually has the smallest computational time for the first two examples, but then has a huge computational time for that third example, which is due to the fact that the moment matching method actually reruns the health economic model. So the more complex the model, the much more kind of the much worse the computational time is for the moment matching method. And the Gaussian approximation method is actually kind of pretty stable across the different examples, and is the worst in the top and the best in the bottom, even though the computational time is pretty much the same. So just to give some quick conclusions, I'm doing pretty good for time. We can just say that all of these methods are accurate in kind of real world models and data collection exercises, but that we couldn't find a systematically superior computation method that we would recommend kind of everybody uses as the default. And actually the trade-off between accuracy, precision, computational time, and ease of implementation will change depending on the health economic model, the proposed trial that you're interested in, and actually the analyst's expertise themselves. So are they more confident using kind of regression methods, in which case maybe the regression-based method is the best. Whereas like the importance sampling method requires you to kind of code up a likelihood. So if you're more confident with coding, then that might be something that you would be better at doing. So as I alluded to, Convoy are actually continuing to work on improving methodology and implementation. We have courses, we have like lots of kind of interesting projects in the work, and we're really trying to make this stuff more easier to do. And the methodological advances that I've spoken about today really facilitate our ability to implement and use value of information in trial design. And really, I think that's the next kind of frontier of this work, is taking some of this methodological work that we know works, we've confidently assessed, and really translating that into practice and designing studies and trials using these methods. So I've actually done pretty well. I've got about 10 minutes left for questions, so I'm happy to take any now.
Okay, so thank you very much for that, Dr. Heath. I'll just check the questions box. Nobody has any questions yet. So if people have questions that they want to put in the question and answer box at the bottom of the Zoom window, you'll see the Q&A bubble. While we wait, and apologies if I missed it, there was a lot of information in there and much of it new to me on the methods. But one thing I think a lot of decision makers are wrestling with at the moment is designing studies for RWE, so real world data and evidence generation post adoption of a technology. So are these methods going to be applicable in that context as well? Or you have a knowing grin there?
Perhaps? Yeah, I don't know. It feels like I set this question up. Yeah, so we actually, I just got awarded a CIHR grant to look at this and to use value of information in this space. So the methods themselves are definitely applicable to designing real world data collection and, making, doing this kind of, I guess, like access with evidence development, like working out the right, the right evidence to collect, the right time to reassess the decision, and all of these kind of key concepts. That comes, I guess, with a caveat of like, methodologists often say, oh yeah, we can do this. Definitely we can do this, but I think that there are a lot of like technicalities and tweaks that need to kind of go into how we do that, that I'm hoping to kind of tackle in the next few years.
Okay, I mean, you didn't ask me to ask that question. I'll just confirm for everyone, but I think we'd probably be really interested to hear about your progress as you go along as well. It's something that as an HTA body at IHE, and there's folks on the call from Calgary who also support HTA work for the government of Alberta, and I think I saw people from INES as well. I think there's a lot of interest in that at the moment, and we'd be interested to see how that goes. So there's a question from Deb Marshall, and it's one that is about EVSI results. So to what extent are the EVSI analysis results being used by decision makers at this juncture?
So I think if we're honest, they're not being used at the moment very much. There is more, like people are more using other value of information measures that are more to do with like what maybe kind of targeting towards outcomes rather than specifically designing the trial. But I think like you probably saw on the dates, like these methods are not kind of that well. I mean, they were developed like relatively recently, and the comparison was published in 2020. So I think like we could never, people have been kind of suggesting that people do this for a long time, and because of the computational difficulties, it never got done. And I think now we've kind of tackled that computational issue with satisfactorily. I think the kind of advocacy side is what we really need to, what we're kind of working on now in terms of getting into kind of decision-making agencies and stuff. So the project that I was just talking is like alongside CADIF. So the goal is to kind of help and alongside the kind of provincial agencies. So trying to kind of work out what the barriers are and facilitators in that space, and really, you know, do the advocacy to get these methods being used in 3, 4, 5 years from now.
Okay. Deb has a follow-up question about whether there's been any research that looks at whether or not the EVSI estimate was sort of correct, you know, and what would be the correct timing to do that sort of research. So when would you expect the EVSI would be realized?
Yeah, so it's a really great question, and it's something that people have like asked me a lot. And the challenge with it is that when we're talking about value of information, we're talking about like the expected value of information. So it's basically saying like across all the possible studies that might happen given the current evidence, what's the value of like information? And when you do the study, you actually only do one realization of that study. And so in a lot of cases, you might find that, oh, actually, I didn't change my decision after the study. So it quote unquote, like wasn't valuable. But what you'd actually need is not just like to do it after one study. You'd actually have to kind of have a portfolio of like studies that you design in this way. And you show that like over that portfolio, we actually did see like a few studies where the decision changed, and therefore like making decisions in this way is better than kind of in the way that we were thinking about it previously. But it's a hard thing to kind of do just based on one study.
Okay, thank you. So another question here from Glenn Sumner is EVSI intended for pre-study design, or can it also be used to post-hoc analyze studies? So clinical trials, for example, or, you know, is it a method to critique existing studies if data is made available?
So it's pretty much a pre-study design methodology. So it's calculating like what's the benefit of doing the study. So I do know some people who've kind of looked back, like said, imagine if I was in, you know, 10 years ago and I had this information, how would it have changed my decision? And they do kind of critique the design that was done. So they might say, oh, if you'd used value of information, we could have saved, you know, 300 patients in this study or something like that. But it's always about like the study, the way that the study is set up. It can't like tell you whether like the data was, I guess it can tell you whether you collected the wrong data, but I don't always know how helpful that comment is to somebody. Like, you've spent 5 years of your life doing this trial, and you could have done it better if you'd collected this data. So it's not something that I've really done, but it can't really, if you have the study data available and you analyze it, that should go into the uncertainty, the evidence, and the decision model, and the value of information should say, what data should I collect given that I've already collected this trial data? I don't know if that makes sense.
I think it does to me, but maybe Glenn can let us know in the comments if he has any follow-ups. Deborah's follow-up is to say thank you for your response. So any other questions? Oh, there's one from Sasha. Sasha von Katwick. So it strikes me that EVSI would be extremely valuable to setting funder priorities, as well as giving a more concrete answer to the infamous "more research is needed" line in the discussion section. True. Is it feasible for EVSI to be employed fast enough and at a scale that would let funders and decision makers set research priorities?
Great question. So I think that's the hope and the goal. I know that there are some people at York who are working with the NIHR. I think that's what it's called. Oh my God, I left the UK so long ago. I don't even know what the funding agencies are called.
Yeah, NIHR.
NIHR. And they're working to do kind of an approximation to some of these things. So the actual complexity of EVSI is not the EVSI part. It's actually often the decision model that you kind of need in order to do these analyses. So if you've done a decision model for whatever reason, then the EVSI can be kind of fairly easily deployed after that to kind of give funding priorities. But often in kind of clinical trial papers, for example, where they may say more research is needed, they haven't actually built that decision model. So they then kind of can't do the EVSI. So I think there's some people are working on how to kind of do it maybe without building this explicit decision model, and some people are working on what a lot of the stuff that we're working on is like the coding and the software and making sure that we simplify the process of these computations as much as possible so that it can really be something that's more feasible for funders to look at.
Thanks. I wonder if there's a role here. A lot of trials have sort of feasibility or pilot phases, and I wonder if we should be pushing for more resources to then do the value of information at that point. I'm not sure trialists will be that happy because we may say that it's not worth going on to the full study, but funders might.
Yeah, I know a couple of people who have been doing that kind of work. And that is something that they have been kind of, that they thought was useful. Actually, most of the time that value of information gets used or EVSI gets used is kind of in the situation where the traditional trial is too complex or too expensive. And then somebody's been asked, like, can you please find a way to like make this less expensive and less complex? And that's where value of information's really been like sold as a solution so far. And so traditionally, I guess, like the 3 times that I've seen it done, it's kind of been used to maybe like reduce the sample size or something, broad compared to like a standard frequentist power calculation. But yeah.
Okay. I see it is now, by my computer anyway, 1 o'clock. So I would just like to wrap up by saying thank you to Dr. Heath for joining us today and giving a great presentation. I can't see the clapping option on my Zoom at the minute. Maybe it's because I've got a presenter privileges. But so thank you very much for that. We really appreciate you taking the time, and I hope we'll be able to hear you talk about your RWE work in good time. And thank you to all for joining us, and I hope to see you again at the next NOAA rounds in about a month's time. So thank you very much.
Yes, thank you for having me. It's been really fun.
Right. Thank you.
Thanks.
Bye-bye.